Now we're going to work on some cytogenetics problems. What I've drawn on the board here is meiosis in a individual that's heterozygous for a paracentric inversion. The reason we can tell it's paracentric is because centromeres here, denoted by these white circles, are outside the inversion loop. So the normal homolog is on top, and the one with the inversion is underneath, and there's a single crossover between genes E and F. So our normal gene order is ABC, centromere D, E, F, G, H, I, J. And what we're going to have to do is figure out how this separates into the gametes during meiosis, and you have to tell me whether these gametes are viable or inviable. So starting at the top, I like to number each of the chromatids so that we get less confused that way. So we'll call this one number 1. And so we just follow it along like it's in a roadmap or something. Genes A, B, C, centromere D, E, F, G, H, I, J, and we end at the top on the other side. So then this number 1 will be to move the computer a little bit. And so we can see this first chromosome has all the genes in the right order and a single centromere like it's supposed to, so it is viable. Now, I'm going to drop down for the next. Try backing this up a bit so I don't have to move it so much. So the next one down we'll call 2. And so we follow the same. We follow the chromatid like this. Now when we get to the crossover, we have to follow it and go on to a different chromatid, like this blue arrow shows, and we come around like this, like this, and we end up on the same side on the bottom. And so this is number 1, number 2 then. It's a dicentric chromosome. So its gene order is, so I'll have to back it up a little further even. So its gene order is ABC, centromere D, E, F, G, D. Second centromere is CBA. So this is a dicentric chromosome, and of course, it's got some deletions and duplications. So gametes getting this chromosome would be inviable. Write it there so that it's more visible. Okay, so we're halfway there. Now let's do number 3. So we start here, genes A, B, C, centromere D, G, F, E, come around H, I, J, and we end up at the same level on the other side. Okay, so this one has a single centromere, and it's got all its genes, although it doesn't show the inversion, but it's still considered viable. We because if the inversion made the organism sterile, it wouldn't be going through meiosis in the first place. Basically, anytime we don't go through that crossover, we'll end up with viable gametes. When you do go through the crossover, that's when you get messed up or inviable gametes. And so now just one left. That one 4, and we go J, I, H, G, F, and where's my chalk? And then we've got to go down like that onto that different chromatid. F, E, H, I, J. And so this one has no centromere. It's an acentric fragment with deletions and duplications. So it's obviously going to result in inviable gametes if they include that chromosome, which I guess they won't because it doesn't have a centromere. So anyway, these are the results from a paracentric inversion. So you'll be given the loop here, and you've got to come up with this stuff on your own. And you'll also have to tell me what kind of inversion. So in a pericentric inversion, which we'll do next, the centromere is in the inversion loop. So let's do one like that now. Okay, here is a inversion loop from a pericentric inversion. We can tell it's pericentric because the centromeres there given by the white balls are in the inverted segment. They're in the loop itself, and here we have a single crossover between genes F and G, and we do it the same as we did before. Number 1, genes A, B, C, D, E, centromere F, G, H, I, J. Results over here. And this is the normal chromosome with all genes present in the proper order and the proper copy number. And so that is going to be considered viable. Number 2, A, B, C, D, E, centromere F. You hit the crossover, and we've got to follow it down to the next chromatid below, and genes G, and then DCBA. And so we end up like before at the bottom of the same side. And so 2 here. And you can see that this gamete will have a chromosome with HIJ deleted and A through D duplicated. So of course it will be inviable. So now we're halfway there. Certainly hope you can see everything. There we go. All right. Let's do the third chromatid. A, B, C, D, G, F, centromere E, H, I, J, and we end up at the same level on the other side. And this one has the inversion, but it's got all the genes in the right number. So it's a viable gamete. And then our last one here, 4. Start here, genes J, I, H, G, F. But we have to follow the crossover and go down to that one. So G, F, centromere E, and then around HIJ, and we end up on the same side at the bottom there. And this one has genes A through D, deleted, HIJ duplicated, so it's going to result in an inviable gamete that inherits that. So this is why we call it semi-sterility, because only half the gametes are viable, the other half are not viable. And that's all we're going to do with inversions. And next we'll do a translocation heterozygote and see how that works out. All right, I'm back, and here's a cross-shaped double tetrad from an individual that's a translocation heterozygote. So we have the submetacentric chromosome with the pink centromeres with genes A, B, C, D, E, F, G, H on it, and the tetracentric chromosome, the blue one, with genes 1, 2, 3, 4, 5, 6, and then the 2 chromosomes with the translocation. So remember, there's 3 different ways this thing can split up, and we'll look at each of those. First, we'll look at alternate segregation. So here the diagonal centromeres go together, so we'll get, see, both normal ones going together, and then both translocated ones going together in the other half of the gametes. So let's look at that. Draw out the results. So these are half the gametes that have both normal chromosomes. So of course they are going to be viable. By the way, if this was going to be on an exam or something, I would supply you with this. You have to come up with this. And the other half of the gametes, so even though these have the translocation, both all the genes are present and there's no deletions or duplications, so these are all also viable. So that's alternate segregation. That's the only way out of a translocation heterozygote that you can get viable gametes. Let's look at the others now. Okay, so one segregation here, the non-homologous centromeres that are next to one another go together. So that would mean we're splitting this thing from top to bottom along this axis here, this dotted line. So these two go together, those two will go together. So I'll put a 1 there for adjacent 1 segregation. So let's do what's on top first. Got our normal submetacentric chromosome, genes A, B, C, D, E, F, G, H. So half the gametes will get this pair of chromosomes. And the other half get this pair of chromosomes. So each group of gametes gets 1 normal chromosome, but 1 translocated chromosome. So there are duplications and deletions here. So these gametes would be inviable. So got inviable gametes there. So So now we've got one more way to break this thing up, and this is the rarer adjacent two segregation where the homologous centromeres go together. So the two blue ones will end up together, the two pink ones will end up together. And that's rare because meiosis is not supposed to work that way. But let's look at the results. We'll put them here. Oops. So here we have the two pink centromeres together, the two blue centromeres together, and of course this has duplications and deletions, or in other words, an imbalanced gamete, so it's inviable, and the same thing for this one. It is also inviable because of deletions and duplications. So this is adjacent to segregation, which again is rare because the homologous chromosomes are not supposed to end up together in meiosis. They're supposed to be separated in anaphase 1. And that is all we have for our cytogenetics problems.